So let's move on to the next question. A 45-year-old woman presents with ovarian tumor. Immunohistochemistry shows napsin A positivity, SNF1 beta, and P53 wild type. WT1 and ER is negative. Genetic testing reveals ARID1A loss. Most likely diagnosis is high-grade serous carcinoma. This is wrong because we already know that P53 is mutated. Clear cell carcinoma is the likely diagnosis. Low-grade serous carcinoma is also not correct, as we will discuss just down. And mucinous carcinoma is also not correct in this case. So the correct answer is clear cell carcinoma, which is positive for napsin A, SNF1 beta, but it is negative for WT1, ER, and it has wild type of P53. And also ARID1A loss is classic for clear cell carcinoma, and in this case endometriosis is a very known precursor of the clear cell carcinoma. So in case of high grade we have WT1 positive. Similarly low grade also have WT1 positivity, and there is wild type of P53, but these markers are negative. In case of mucinous, it is CK7 and CK20 positivity, but lacks all other markers. So from this we conclude that serous tumors are WT1 positive, and to distinguish between low and high grades, low grade are often wild type P53. So now, using the same knowledge that we studied just now, let's solve the second question. 38-year-old woman with ovarian cancer has IHC showing WT1 positivity, which points us towards serous, and P53 is wild type, so it might be low grade, or P53 signature below lesions below the P53 signature. So another thing is it has ER negativity, and KRAS mutation is specific for low grade serous carcinoma. So the likely diagnosis is low grade serous carcinoma. This is wrong. Clear cell carcinoma is also wrong because it is WT1 negative that we just studied. And endometrial carcinoma are ER positive, and they often consist of CTNNB1 or P10 mutation instead of KRAS, which is specific for low grade serous carcinoma. So let's quickly discuss different precursor lesion of low grade serous carcinoma. So it develops from the tubal epithelium, which then give rise to ovarian epithelial inclusion, and after that it will give rise to benign cystadenoma, and after that we have borderline serous tumor, and ultimately it gives rise to low grade serous carcinoma. And as I have already discussed, low grade serous carcinoma does not give rise to high grade serous carcinoma, with the exception of P53 mutation, which might give rise to high grade, but high grade often arises de novo, and we have already discussed this in the first part of our series. So different mutations playing role in low grade serous carcinoma include KRAS or BRAF and ERBB2. These are all specific for low grade serous carcinoma, not high grade. We have already discussed genetic predisposition for high grade. Also certain allelic imbalance like 1P, 5Q, 8P, 18Q, these results in low grade serous carcinoma.